Good evening, everyone, or at least good evening for those of us on the East Coast. I'm Dina Matos. I'm the Executive Director of CARES Foundation, and we are pleased to bring you this very informative program today in conjunction with the Adrenas Therapeutics. Adrenas is a subsidiary of BridgeBio. BridgeBio is a company that focuses solely on rare diseases, and Adrenas is developing this therapy for CAH. So we're super excited to have you join us tonight to learn about the potential for gene therapy for CAH. I just want to remind everyone that we will have the opportunity for Q&A. So we're going to ask you to use your Q&A function to send in your questions. So the first half, we'll have a presentation, and then we'll allow plenty of time for you to ask questions. So feel free to go ahead and ask them when the time comes. Now I want to introduce you to Dr. Eric David, who is the CEO of Adrenas. We've been working with Eric and Adrenas since about 2018. It's been a wonderful partnership. They are invested in this program for CAH gene therapy, and they've been a great partner, listening to patients and getting to know, what their struggles and challenges are, and what the hopes are for this therapy, which is, I guess, the closest thing to a cure for CAH. So without further ado, I want to introduce Dr. David. He is no stranger to innovative therapies. He's been with Adrenas for several years, but has significant experience in this area, and has worked in a lot of different areas, including a practicing physician. So, Eric, take it away.
Thanks so much, Tina. I really appreciate it. And good evening to all of you, or good afternoon to some of you on the West Coast or elsewhere. And thank you so much for joining us tonight. As Dina mentioned, I'm the CEO of Adrenas Therapeutics, and we are so proud to be partnered with CARES. As Dina mentioned, we came to our first patient meeting with CARES back in 2018. And BridgeBio is indeed, as Dina mentioned, very focused solely on rare diseases. And the group on this call works exclusively on gene therapy. And we have a couple of clinical stage programs right now, and this one for CAH is one of them. And we're so excited to be able to speak to you about it tonight. We have learned so much from people living with CAH and from the CARES community. And some of the folks on this call who you'll meet, this is part of a team that works just on our gene therapies. And this is, there are a whole lot of other people who have worked on our gene therapy for CAH, everything from manufacturing to all the early studies in animals and the basic science research. But the group here tonight is really focused very much just on the clinical side, on this clinical trial. And so we have with us today Kathleen Kirby, who heads our clinical operations. We have Adam Shaywitz, who is our chief medical officer. And we have Kamal Barucha, who is the vice president of clinical development. And so our goal here tonight is really just to focus, spend most of the time, hopefully, just answering questions. And so we didn't want to just be talking at you guys for an hour. So we'll aim to spend just about 20 minutes or so as a basic introduction, hopefully answering a lot of the questions that we know folks have about what is gene therapy, and, is it safe? And tell us a little bit about the clinical trial, questions that we know a lot of folks have. And we'll aim to cover that in about 20 minutes or so. So as I mentioned, we've learned so much from the community in all this time. And we also have, spent a lot of time speaking, not just with patients within the CARES community, with folks living with CAH, but we have also spent quite a bit of time talking to some of the clinicians, several of the clinicians who are at the care centers of excellence. And so, we've been we've been very fortunate to learn from people living with CAH about all of the tremendous unmet need in the disease, and the fact that it is, it is something where CAH has had, as so many of you in the community have pointed out to us, CAH has had, very little innovation in terms of the therapies available in the last several decades since the advent of steroids. So that was one of the reasons that really brought us to become interested in CAH for gene therapy. I think the thing that the thing about gene therapy is it really aims to take the gene that is not functioning properly in people living with CAH and simply replace it so that with a one-time infusion, there's the potential for people with classic CAH to make their own cortisol and aldosterone in the right amounts and at the right times. And no other therapy can really offer that, because the ability for one's body to make cortisol at the right amounts and at the right times it really means that it's the only therapy out there that can offer the potential for people living with CAH to completely get off steroids or very significantly reduce their steroids. And so that's what excited us so much about the disease, as we learned as we learned from people living with CAH. We just thought this is a perfect candidate for gene therapy. And the very basic question of what is gene therapy, I'll turn it over to Adam in a moment to and then to Kamal to talk everyone through what gene therapy is and why we believe that CAH can really be treated. That gene therapy has great potential for people living with CAH. When we talk about trying to put in a functional copy of a gene that is not working properly in people living with CAH, the question always comes up: how does one get a gene into the body, and not just into the body, but into the cells that we want it to get to in the adrenal gland? And when one asks the question, well, how do you even just get a gene into the body? There's an answer that, there's something out there that has been doing that job of taking a gene and putting it into our bodies for millions of years, and that's viruses. That's exactly what viruses have evolved to do over millions of years. And so, as the team will tell you, what we do in gene therapy is we take a virus that is not known to cause any human disease, and we take out the DNA that virus has, that usually it delivers its own DNA into our bodies, and we replace it, in this case, with the 21-hydroxylase gene. So again, very excited to tell you a bit more over the next 15, 20 minutes, and then to answer your questions. So with that, I will turn it over to Adam.
Great. Thanks so much, Eric, and thanks, Dina, for organizing this. So as Eric mentioned, I'm Chief Medical Officer. I'm also an endocrinologist by training. And this approach, as an endocrinologist is really exciting. And it's really exciting because, to kind of continue what Eric was saying, our therapy, it's not a dose of hydrocortisone. It's not a dose of prednisone or dex. When we infuse our therapy into a person with CAH, we're going to be not infusing a dose of a glucocorticoid. We're infusing essentially the potential for the adrenal gland to function properly. So in some ways, it maybe the best way of thinking about this approach is it's really like giving somebody back a functional adrenal gland. And that's what the gene therapy approach really at its best can do. So if you've been infused with something that essentially can give you back a normal functioning adrenal gland, what does that mean? That means that you can make cortisol a lot when you need it. When your body is stressed, if you're sick, you make a lot of it. If you don't need it, your body will shut it down naturally. It'll be responding to your body's normal rhythms. Your brain, as many of you know, controls a lot of the cortisol response. If you don't need the cortisol, your functioning adrenal gland won't make it. Same thing for aldosterone. So for those of you who are on Florinef, if you now have a functioning adrenal gland, you won't need to take Florinef. And your body will make aldosterone when you need it, and it'll shut it down when you don't. So what does this mean? It means that you don't have to take higher dose glucocorticoids. You don't have to be on a lot of Dex, a lot of prednisone, and in doses that aren't really natural, and you won't have the side effects of things that some of you are already well aware of. You're taking a lot of hydrocortisone, prednisone, Dex, and you can't mimic your body's natural rhythms. Your body will have the side effects from that, weight gain, diabetes, metabolic issues like that, problems with sleep, bone issues. On the flip side, when you do need it, you'll be able to make it. You'll be able to make a lot of it. You may even not need stress dose steroids. So if you're someone who has CAH, or if you're a parent of a child with CAH, you worry about that a lot, especially with your kids, about getting sick and about not having enough cortisol around. And if this therapy works to the potential that we hope it can, then you'll be able to make cortisol. Your child will be able to make enough cortisol when they need it, but we'll shut it down when you don't. So the best way, that's probably the best and easiest way to think about what our therapy is. It's an infusion of the potential for normal adrenal gland. It's not an infusion of a dose of any glucocorticoid. So Eric mentioned this, and this is, and he really nicely went through this, and this is a visual depiction of how are we able to kind of infuse something that will give your body the potential to have normal adrenal gland, normal adrenal function. And I know some of you may know this already, so just bear with me as I sort of walk through sort of some of the basics. The CAH is similar to a number of diseases in that there are a number of diseases where your body can't make the right protein. It's either missing or malfunctioning protein. And if in those kinds of diseases, if you can give back that missing or dysfunctional protein, you can have significant impact, potentially even cure the disease. So with CAH, the missing or dysfunctional protein is called 21-hydroxylase. And the instructions in your DNA that tell your body to make 21-hydroxylase is a gene called CYP21A2. Why it's not called 21-hydroxylase or 21-OH, I don't know. There's a history of how genes are named and proteins are named, but the CYP21A2 is the instructions for making the missing or defective protein 21-hydroxylase. So if you can restore, if you can give your body back the missing instructions or the corrected form of the instructions, those instructions can go into your adrenal gland and tell your adrenal gland all the time, make correct 21-hydroxylase, make the correct 21-hydroxylase, and then you'll be able to make cortisol and aldosterone at the right time and at the right place. How do you do this? Well, you can't just give the instructions. You can't infuse the instructions. It would just get destroyed in your bloodstream. So you need to surround the instructions with a sort of delivery truck, a kind of you sort of think about it like a sort of hard eggshell, and instructions are like the yolk. So you put the instructions, the 21-hydroxylase coding sequence CYP21A2, into a vector, the AAV5 vector that Eric told you about, which is a vector that is part of a virus that many of us are normally exposed to and have no problems with, and don't get any problems with. We don't even know that we may have been infected with that. So we put that gene into the delivery truck, the AAV5 vector. That gene, the CYP21A2 plus the vector, we infuse that through a kind of like if you dehydrate it and you get an IV infusion, it will go into a vein in your arm, be distributed in your body, and go to your adrenal gland. And it will essentially, these delivery vehicles will dump the correct form of CYP21A2 message into the adrenal gland, into the adrenal cortical cells. And now the adrenal cortical cells will have a functioning set of instructions for making the right form of 21-hydroxylase, and that will allow your body to make cortisol and aldosterone at the right time at the right amounts. And all of this, this has been a system that's been worked out over decades. And we have our own data that shows that this in fact does happen. We can get 21-hydroxylase and the message for making 21-hydroxylase into adrenal cortical cells. So as I mentioned, there's that system of the adeno-associated virus and the message, and using that to deliver missing or defective proteins, the correct form of missing or defective proteins, is a system that's been used for quite some time now. So AAV gene therapy, even though it's new to us in the CAH world, it's really not a new form of therapy. It's a really exciting form of therapy because it offers the potential to do what I described, to sort of have a functional organ or tissue. But it's really not new. There have been, over the past few decades, that AAV gene therapies have been used. There have now been 3 approved AAV gene therapies in the US and in Europe. Over 100 active or completed AAV gene therapy clinical trials, and thousands of people, over 3,500 people, have been treated with AAV gene therapy. So there's a lot of experience here. And AAV gene therapy in clinical trials has been around for several decades. Similarly, the adrenous sort of therapeutics, the idea for using this for the AAV gene therapy approach for CAH has been around for about a decade. The idea that our company was sort of founded upon was sort of born from work of a pediatric endocrinologist in France named Pierre Bonnier. And about a decade ago, Pierre did some very exciting experiments in his lab, looking at the way of using AAV gene therapy in a mouse model of CAH. And he showed at that time, about a decade ago, that AAV gene therapy could essentially have significant impact in the mouse model of CAH. Since then, we at Adrenas have partnered with Pierre, sort of taking the baton from his group, still collaborating with him closely, but optimizing the AAV gene therapy and doing additional studies that allow us to move into the clinic. So over the past 4 or 5 years, what we've done in collaboration with him, but a lot of work done by our own team as well, is optimizing the gene therapy to the form that it's now in, which is called BBP-631 is the name of our AAV gene therapy. And we've done additional animal studies that speak to the safety of this in rodents and in monkeys. And we've also shown that the idea that I showed a couple slides ago, that the vector plus the information, the genetic information, would go into the adrenal gland. We see that in monkeys. We can see that IV infusion of the gene therapy does go into adrenal cortical cells in a monkey and makes enough form of the right 21-hydroxylase protein and functioning 21-hydroxylase protein, enough of it that we strongly believe that can make a clinically significant impact in people with CAH if we can repeat that in what we saw in monkeys in people. Just this year, we've reached some major milestones. The FDA looked at our whole set of experiments and safety in animals, in potential efficacy in animal models, and looked at the way we were making the drug and gave us the green light to proceed with clinical trials in people. So our investigational new drug, or IND, was cleared to proceed this year. And we were also granted fast track designation by the US FDA, which basically acknowledges that there's significant potential for our gene therapy approach to have an impact in CAH. So with that, I'll hand off to my colleague, Kamal Baruka. Kamal.
Thank you, Adam. Adam and I work very closely together, and as he's trained as an adult endocrinologist, my training is as a pediatric endocrinologist. And this slide just introduces the umbrella of our CAH gene therapy program, with one adventure arm for treating adults with CAH and the corresponding program for children called ADREAM. And we've put up a very, we believe, informative website where there's also answers to frequently asked questions, and I think also an opportunity to ask additional questions. So although this is just kind of giving an overview of what we have, in terms of layout of our program, there's more information behind these logos online. And as Adam alluded to, we're starting our first studies in human based on the shoulders of very robust safety model organism data and manufacturing standards that the FDA has cleared for us to proceed. And we have we are now enrolling, we're excited to say, a adult trial, and Kathleen, the next speaker, will go through some of the mechanics of getting involved. But we're actively enrolling an adult early first program whose primary goal is to identify a safe and effective dose. And upon initial safety and a sign that the approach is working, we hope very quickly to initiate pediatric studies in close collaboration with the regulators to devise an appropriate time on that we can safely enter the children with classic CAH. And every patient and every participant that is dosed in our program will be followed for long-term safety evaluations and durability. And this is just a very general overview of our core program, which is the first in human study in adults. As you can read through the chevron, on the left, there'll be a 1 or 2 month screening period where you will be assessed with both blood and other evaluations to see whether you qualify for proceeding to the dosing step, which is summarized as a baseline part of the program where you will actually get the one-time infusion of the medication or the gene therapy. And unlike other programs, it's a one-time intervention, but there is rigorous follow-up for efficacy, which means whether the drug is working or not, or the intervention is working or not. And a lot of the evaluations we'll be doing, as you can imagine, will be trying to assess how effectively your body is making cortisol, in addition to some of the traditional markers many of you may be used to, such as 17-OHP and A4. And then the tail end, as we alluded to, there'll be a long-term follow-up period where we'll be following in less frequent and more spaced visits along the way. And we've tried to make the overall trial adaptable as possible in terms of having flexibility and home assessments, just sensitive to just everyday work and life commitments. But at the same time, trying to maintain some sort of rigor in how we can evaluate some of the exciting data that we hope to collect. And I'll like to hand off here to my colleague, Kathleen, who will cover the next couple of slides.
Thank you, Kamal. So my group, as Eric introduced early on, is development operations, which working very closely with Kamal, Adam, and several other of our team members, oversees the day-to-day running of the clinical trials. And so Kamal very nicely introduced our overall advanced program, and then specifically laid out plans for our adventure trial. And what I'd like to talk with you about are some of the key eligibility criteria. So we're looking for people who are 18 years of age or older, excuse me, who have classic CAH due to 21 hydroxylase deficiency, and that's either the simple virilizing or the salt wasting form. These people do need to be on a stable oral hydrocortisone dose as the only glucocorticoid therapy. You can find other additional eligibility criteria on clinicaltrials.gov, and I believe the link to our entry on clinicaltrials.gov will be placed in the chat in case you'd like to click that link. So one key feature about our program that we realized was that while we picked great investigators at great sites, not everyone lives near one of those centers. And so we established a pre-screening effort, and I'd like to just walk you through at a very high level the enrollment steps. So you would visit cahgenetherapy.com, and as you can see on the slide, you'll find a button where you can either click to stay connected, or you're able to continue through a pre-screening questionnaire, and it asks a few simple questions. And if you are answering in the positive way, then you'll be asked for some contact details. You'll hit submit, and then one of our team members will be calling you to conduct a little bit longer interview, asking you for some medical history and your medication, your current medication status. And so if you, again, work your way through those questions and you are potentially eligible for our gene therapy trial, you will be referred to Science 37, which is a clinical site that conducts virtual trials. They will walk you through an e-consent process, and then what they'll do is schedule an at-home eligibility assessment. They will draw some blood, and then through that testing, we'll be able to determine if you're potentially eligible. And then as a space becomes available in our gene therapy trial, you would be referred to a center, and you can then take steps to proceed into the full eligibility for our study, for the gene therapy study. So what I'd like to highlight here are, again, the website that we've designed for the CAH community, cahgenetherapy.com. If you have specific questions, you can write to the email address that's shown. And again, I believe it will be posted into the chat. But I would like to say that participating in a clinical trial is so important, because behind every new treatment are the people who volunteer to participate in these studies. And we're grateful to the patients who do, and just want to say thank you for that. So I'd like to now turn it back to Dina for our Q&A.
Thank you, Kathleen, and thanks to Eric, Adam, and Kamal. I know it was a short period of time, but we covered a lot of, great information, but we have some terrific questions. So I'm going to read them and then we'll get them answered for you. So number one, how can the new functioning gene be the same for both salt wasting and non-salt wasting? Adam or Kamal?
Sure, I can take a crack at that, Dina. Thank you, and thanks for the question. So for both the salt wasting and non-salt wasting forms of classic CAH, the great majority are due to of both forms are due to missing or defective versions of the CYP21A2 gene that codes for the 21 hydroxylase enzyme protein. So we would want to replace the same gene for both of those. So 95% or more of classic forms of congenital adrenal hyperplasia are due to problems with 21 hydroxylase protein, CYP21A2. So it's the same underlying gene that's missing or defective for both.
Great, thank you. So the next question is about whether it's more effective in the younger population or older. Does age make a difference? And is this considered a fix or a cure?
I can take that one or first crack. We have reason to believe that there'll be a prospect for benefit in both children and adults based on ongoing results in other trials with gene therapy. Some of you may know about a devastating illness called spinal muscular atrophy. There's actually approved AAV-based gene therapy for that condition. And some data in other conditions such as hemophilia, whose trials have been conducted in adults, have also shown signs of positive benefit, although those trials are ongoing. So we have reason to believe that both adults and children would be potentially good age ranges to help.
No, that's exactly right, Kamal. And you know, I think that there's also, as part of that question, I believe the whoever asked it was interested in understanding, you know, do we know how long this will be effective for, whether the person with CAH being treated is young or older. And the truth with gene therapy is that at this point, we don't know exactly how long it will last. We have reason to believe, based on the work that we've done with CAH, that it will last for some time, but we don't know the answer. What I can tell you is that, you know, some of the longest gene therapies that have been dosed in patients were dosed about 10 years ago. And they relate to the trial that Kamal just mentioned. There's a disease that many of you know of, have heard of, called hemophilia. And that is a disease that is being treated with a gene therapy that uses the same delivery vehicle, AAV5, that we are using here for CAH. And some of those patients who received that AAV5 treatment for hemophilia are now 10 years after their treatment, and they still have efficacy. They're still completely off of the drugs that they had previously been taking for hemophilia. And so that's a good that's a good data point for us to see in terms of durability. Now, you know, whether or not gene therapy for CAH will last for any number of years and how many years, we just can't say at this point. What I can tell you, in addition to you know, that data point that I mentioned about this other AAV5 trial with data out to 10 years, I can tell you also that there are so many gene therapies being developed for so many different genetic diseases right now. And this question of durability and not knowing exactly what the durability will be obviously comes up in every single one of these trials. And so it is something that we as a community, all of us who do gene therapy and are working with patients with genetic diseases, this is a question that we know you know, we will have to address, because we want to be sure that if the efficacy does fall off in some patients, that, you know, that there's a way perhaps to give a second dose of it. And so that is something that I can just tell you that all of us are thinking about and actively working on in the background.
Why did you stop using AAV-RH10 as a vector for the gene therapy and used AAV5 for the delivery in clinical trials?
That's a great question. If you can we go back to the slide that had the pictures of the screenshots of those academic papers? That slide has just a photo of the original paper that our scientific founder, Pierre Bunière, had published several years ago. Here we go. You see this paper on the left side of the screen that says an AAV-RH10 vector. So Dr. Bunière, who was the first one to publish on gene therapy for CAH, he was a pediatric endocrinologist in France, in Paris, who had been practicing and treating CAH patients for about 30 years. And he was the first one to develop this gene therapy. He used a different, as I mentioned, we use in our trial AAV5. He, in those very early studies, used AAV-RH10. he selected it simply because it happened to be one of the easier ones that he had access to. And so in that paper that you see on the left side of the screen, it was AAV-RH10 was great for proving the general concept that CAH could indeed potentially be treated very effectively with an AAV gene therapy. And when Adrenas acquired that program from Dr. Bruniere and started working with him and taking this program, getting it ready to take into the clinic, we had to ask the question, what would be the best AAV subtype? Because there are many different types of AAV. What would be the best ones for CAH? Because we need something that could get very effectively into the adrenal gland. Not all AAV types go to exactly the same tissues. They have different specificities based on their sort of the cells that they tend to prefer to go to. And so we tested a number of different AAV types before we selected AAV5. And the reason we ended up selecting AAV5 and taking that forward into the clinic is that AAV5 very effectively gets into the adrenal glands. And then it also has, it's unlike RH10, AAV5, the one we selected, is one that has been in the clinic. As I said, now there have been hundreds of patients treated with AAV5 for another disease, for hemophilia. So that was another thing we really liked about AAV5, was that it had been in the clinic in patients previously for a different genetic disease.
Thank you, Eric. Next question. So this individual is asking, appears that PubMed indicates that recruitment is only taking place at NIH in Maryland, and this person lives in San Diego. When will they be recruiting in California or Southern California? Kathleen, do you want to take that? I'm not sure.
Sure, Dina, I'll take care of that. So what I'd like to offer a clarification. If you visit clinicaltrials.gov, you'll see the clinical sites that we are working with. Currently, the University of Minnesota is our open clinical site, which means they've gone through the requirements of getting the appropriate approvals and submitted the documentation. And so they are currently the one site that is open for enrollment. We are working with NIH. It's a lengthy process to open the site there. And so we're working with them and have regular contact with Dr. Merck and her team. And we're also working with Children's Hospital LA. So both of them are going through the process. It is a little difficult to say because there are committees they need to go through and many steps that need to be achieved. But staying connected to clinicaltrials.gov is the best way. And we also do send out notices from time to time as a new clinical site would open. That would be an interesting piece of information. We would share that with our advocates as well. So I hope I've addressed the question sufficiently for you.
Yeah, and I would say that, we hope to have some of these other sites open in the not too distant future. As Kathleen mentioned, you can stay in touch on our website. But I should stress something that Kathleen mentioned earlier, which is even though, for example, in this case of someone living in San Diego, even though CHLA is not open quite yet, you can still enter the screening program to be screened at your home. Anywhere in the country, and so that would help determine the eligibility for being dosed with the gene therapy.
The next question is about disqualifiers such as statins or thyroid issues. I know there's lots of information on clinicaltrials.gov/adventure, but I don't know if anyone wants to take a few seconds to address this.
Not all chronic medical conditions are exclusionary. I think we'll handle that on a case-by-case basis, but I can say generally, if you're well maintained and there are no concerns about how that additional condition is managed, it potentially could be allowed to be it could be allowable in the trial, but it's a case-by-case review, so it's hard to answer that definitively without the specifics.
Yeah, and just to add to that, which is entirely true, is that we understand that that adults living with CAH have same set of issues in many ways that people that are living without CAH or adults may have: thyroid problems, hypertension, hyperlipidemia. So the intent is really for this trial is not to require people to be off any medication, but to recognize that there probably are going to be lots of common things that people may be dealing with. And I don't think there's anything specifically exclusionary about those issues that were mentioned: drugs for hyperlipidemia like statins, or if you're on Levoxyl for hypothyroidism and your dose is pretty stable, that should be fine. But as Kamal said, there are things that you'll have to talk to the site investigator about. But if you go to clinicaltrials.gov, you'll see what the sort of main exclusionary criteria are. But the intent is really not to exclude if you're on anything, but more if there's a major underlying issue.
And if there are any questions that people have, again, about that or anything else, because some of the language on clinicaltrials.gov can be confusing, so please do feel free to get in touch with us at the email address on the screen.
Great, thank you. So another great question. So if someone completes gene therapy before having children, will they pass on the faulty gene, or will the body recognize the gene as normal?
Yeah, that's a good question. I can take a crack at this, Dina. And this is a question that's come up many times before. People are, for many reasons, in some ways concerned. Like, this question is, is there a risk of passing this on? Some people are excited about the potential of passing on a functional version of the gene to children. But the answer is really that the way that our gene therapy is designed, it's designed not to infect or transduce cells of your ovaries or testicles. So it's really not designed to go into cells that are passed along to your children. So from a safety standpoint, it's something that's a good feature. It's a feature, not a bug. But unfortunately, we don't believe that if you have the disease and you take this, that the gene therapy would be passed on to your child through normal reproductive processes. But ideally, we would be able to, as Kamal went through, we would be able to move through to pediatric studies very quickly. And the aspiration of our programs to be able to treat very young children too with this approach.
Thank you. Of the clinical trials using this type of gene therapy on other diseases, how successful have they been?
So there's, as I think Eric has mentioned with hemophilia, and Kamal has mentioned with spinal muscular atrophy, there's been a lot of success with AAV gene therapy in general. Some people, there's different forms of that shell, of that vector. We're using a specific shell called AAV5. And there are other shells that have been approved, and AAV5 is actually in wide use in clinical trials right now for hemophilia A and hemophilia B. So we're lucky in the sense that we have a lot of information from these hemophilia studies where the same vector, the same shell, has been infused into adults who also have a disease where they're not making a functional protein. And now we can kind of assess, okay, what are the safety signals that are coming from the AAV5 trials, and what are they seeing in terms of success? So I think there's reassuring aspects from both of those right now. The hemophilia trials so far with AAV5 have shown that at least what's been publicly presented and we're aware of, no significant safety concerns have been presented. We see that the protein that's been corrected is made in the body, and the clinical effects of making that protein persist for years. So with AAV5, it's out to 5+ years. With other forms of AAV that started even earlier, so we have more data, we can see effects out to 10 years or more. And, we don't know if this is what we're going to see, but we have, some hope because it's the same shell. There are some common features of AAVs that allow us to design our trial in a way to minimize known side effects, and that's what we've done as well.
Thank you, Adam. Another question is about the time frame, and we're assuming it's for the overall development and approval. Kathleen, perhaps you can answer.
Sure. So that's a difficult one to really answer. I think what we've laid out for you are the initial steps of our program. To a large degree, it does depend upon enrollment and proceeding with, bringing adults into this existing program and then proceeding on to the next step. So at this time, we really can't give a time frame, but I think it's important to note that this group on this webinar, as well as many people behind us, are working as efficiently as possible. We're constantly evaluating ways that we can creatively approach this program development in the interest of our patients.
Thank you. Another question is about the typical time period to see the clinical effects and do the adrenals shrink to normal size. Adam or Kamal or Eric?
I can try to start. This is the first in human study, so we don't know to be honest, but we're planning the assessments in the order of magnitude of weeks after the infusion, not months. So we are gauging for cortisol production in the sequence of weeks following the infusion, and that's our best expectation for seeing gene expression.
Thank you, Kamal. What are some of the possible side effects of gene therapy? Adam? Sure.
I touched just a couple minutes ago on the experience with this type of gene therapy, AAV5 gene therapy, and how what we've seen mainly from the hemophilia trials, hundreds of patients being treated with AAV5 adults with hemophilia, which is reassuring from both a safety as well as a efficacy standpoint. But there are known effects of AAV gene therapy as a class. One of the known effects is that there often is a very transient but fairly common rise in some liver function tests because the gene therapy often does go to the liver as well as some of the other organs, in our case to the adrenal gland. And so in order to prevent that rise in the liver function tests, most sponsors use immune suppression, some form. They typically use glucocorticoids to make sure that that rise in the liver test is not going to occur. We generally think that the rise in the liver function test is not really a safety concern as much as is something that could affect how much of the protein is made. So in our case, we're concerned about the adrenal gland, and we'd use the liver as a surrogate of what might be happening in the adrenal gland. So we want to make sure that the gene therapy gets in the adrenal gland and it stays there. We don't want it to get there and then the cells that have it are somehow eliminated. So to prevent that and to get sustained gene therapy effect, we often give, and sponsors using gene therapies give immune suppression. Because of the conversations we've had with you all, the patient community, as well as expert physicians who treat patients, adult and children with CAH, we've opted not to use a high-dose glucocorticoid immune suppression regimen. We're using a low-dose form of another immune suppression called tacrolimus in order to avoid high-dose glucocorticoids, because we understand that you all really, it's something that is not liked by this group for obvious reasons. And there are some other things, some programs, the toddler program for spinal muscular atrophy with another shell, they've seen minor abnormalities in some blood counts right after dosing. Hasn't been reported in the adult AAV5 as much, but we're going to keep an eye on that and would be able to act on it if needed. But I think the main thing is a transient increase in some liver tests that are seen if you don't immune suppress. So that's why we use immune suppression.
Thank you, Adam. If efficacy lasts 10 to 15 years, can the process be repeated? Adam?
Yeah, so this is a question about, can you redose with AAV gene therapy? And this is a really important question for the field because, you know, as Eric mentioned earlier, a lot of these questions are questions that it's not just for us as a sponsor, but the whole field using AAV gene therapy for, you know, hundreds of different conditions is wrestling with. So we mentioned that we want to test if people have antibodies to AAV5, because if you have antibodies, if you've been exposed to AAV5 and you generate antibodies to it, if we were to give you our gene therapy, your immune system would recognize it and say, not in our blood, and get rid of it. So the same thing would happen if somebody who doesn't have exposure to AAV5 and gets our therapy in the context of a clinical trial, you would make antibodies against AAV5, very likely make them. So if we were to try to redose, the same problem would occur and you may not get the maximum effect. But this is something that has been high on the radar of all the gene therapy sponsors, companies. It's something that we feel confident that over the next 3 to 5 years, there will be a clear path forward. Right now, there are multiple clinical trials that are experimenting with different ways to allow for redosing using known techniques as well as experimental techniques to get rid of antibodies. So very likely in the near future, there will be ways to redose. We can't say right now that we can reduce, but we're very confident that over the next few years, there will emerge at least one, likely multiple ways that will allow for redosing.
Thank you, Adam. Another question is about the stable dose of hydrocortisone. What does it need to be? Is it 25 milligrams, 30, or another dose?
No, the stability, all those doses are eligible for the trial. The stability refers to how long that you've been on a given dose. I hope that answers the question.
Yeah, so the amount of hydrocortisone that you're taking is, to Kamal's point, it doesn't matter. We just want to make sure that you've been on that same dose for a little while, that you didn't, for example, just increase or decrease your dose right before starting the clinical trial.
Great, thank you both. Would the success of the trial be binary? In other words, will you be able to determine if it works or doesn't work? When do you expect results? And how would gene therapy mimic the circadian rhythm of cortisol and aldosterone? Adam or Kamal?
I can take the first part of the question, and then certainly Adam and Kamal, as our resident endocrinologist, can comment more. So the because we are able to measure cortisol in the bloodstream, as Kamal, I believe, said earlier, we would be able in a matter of weeks to to do essentially an what's called, many of you are familiar with, an ACTH stim test, where we would have the person who had been treated with the gene therapy come into the hospital for a night or two. We would hold their evening dose of their glucocorticoids, and then we would be able to measure their cortisol, a stimulated cortisol test in the morning, and therefore really determine, is your body making cortisol, and if so, how much? And so we would do that a few times, and if after a patient has had the gene therapy for several weeks, if we did this a few different times and the body was making enough cortisol, we would work, the principal investigator, the treating physician here, would then be able to start to taper the glucocorticoids and just keep making sure via blood tests that the body is making enough cortisol.
Great, thank you, Eric. Let's see. I have a ton of questions. So someone is asking about the time frame. I think we talked about that already. Do you expect long-term therapeutic effect to take into account the rapid tissue generation of the adrenal gland? I think this has kind of been answered, but if anyone wants to add to it.
Yeah, I mean, I know we've sort of talked about this, and you know, I think the bottom line is there's really not a lot of evidence that there's turnover in the adrenal gland after the first few months of life. So, you know, this is an issue. Obviously, a lot of gene therapy companies are very concerned and interested in, okay, we're delivering gene therapy to a particular organ and cells in that organ. Are they going to be turning over rapidly? So companies that are interested, like hemophilia companies in liver-directed therapy, are interested in liver turnover. So one of the first things that we did as a team is think about adrenal gland turnover. Is that something that really happens? What is the data? And when we really dove into the data and spoke to a number of experts, the sort of picture that emerges is that after the first few months, there's no real evidence that there's significant adrenal gland turnover. So certainly in adults, and very likely in children also, down to very, very, very young ages, there's not significant turnover of the adrenal gland cells. So, you know, as you might imagine, it's very hard to kind of prove a negative, but based off of a number of studies in, you know, people, some autopsy studies, unfortunately, but they're informative studies in monkeys, it's very clear that there are differences between primates, like monkeys and people, and rodents. So we feel pretty confident that there's not significant adrenal gland turnover after the first few months of life.
Great, thank you, Adam. How does the AAV know to the adrenal cortex? I guess the question is, how does it know to travel to the adrenal cortex? I'm not sure. Adam or Pamo?
Sure. So I can take a stab at that, and then, you know, Eric, also, I know you've been thinking about this for a long time too. There's a lot of different shells, AAV shells. They tend to have different sort of homing directions. And for example, the approved gene therapy for the rare neurological disease in children called spinal muscular atrophy, that's using a particular shell that goes into the brain and the nervous system when you infuse it into veins. So it crosses the blood-brain barrier. But no one really understands exactly why that happens, but you do the experiments and you see that in rodents and in monkeys, it does go into the brain. Similarly, we were very open-ended about our approach. We evaluated, as Eric mentioned, different AAV shells after the initial studies with AAV RH10, because we wanted to be driven by the data. And what we saw is that AAV5 does go to the adrenal gland in very large amounts. And so we don't have a great answer for why it does. There's likely some surface proteins on adrenal gland cells, adrenal cortical cells, that interact with proteins on the surface of the AAV5 capsid. But we can't sit here right now and say we know exactly why it goes to the adrenal gland. All we can say is that it does, and it does not only in mice but also in monkeys as well.
Yeah, it's a great question, and it's sort of very similar. The answer is really, as Adam said, it's very similar to sort of any other virus, whether you're talking about the virus we use here, AAV, or a cold virus or a stomach virus. You know, why does a particular virus always cause a stomach flu versus another type of virus that always causes a cold or a cough? It's just sort of the cells that virus seems to prefer in a very predictable way. And everything we've seen from AAV5, because we tested several different kinds of AAV to see which ones got to the adrenal gland, And AAV5 very predictably in all of the studies we did went to the adrenal gland very well. And some of the other forms of AAV didn't. So that's why we selected AAV5.
Thank you, Eric. So we've come to the end of our time. We have another 2 questions we're going to try to cover real quickly. So one is, are the adrenals expected to shrink to normal size? Adam, Kamal, Eric, who wants to take that one?
I'll take a stab at that one. I think the sort of current thinking is that the adrenal gland increase is driven in a large extent by the high levels of ACTH. So, if you're not making enough cortisol, your brain and pituitary secretes a lot of ACTH to try to get your adrenal glands unsuccessfully to make more cortisol. But that effect of the high ACTH drives at least some of the increase in adrenal gland size. So if our therapy works as we hope it will, where your adrenal glands are responding normally and physiologically to ACTH, then your ACTH will be lower. It won't be really high because it's trying unsuccessfully to get your glands to make something. It will be decreased and it'll be physiologically, it'll be slightly high when it should be high, but generally a lot lower. So the prediction would be that there would likely be some shrinkage as the ACTH is lowered, but that remains to be seen as we do our studies.
Great, thank you, Adam. How long, and I hope I pronounced this correctly, is the tacrolimus administered? Is it taking long or short term? Kamal, do you want to take that one?
Yeah, I can answer that. Short answer, a very short term. It's going to be administered as a low dose. That's been used safely in other indications to reduce or spare the use of steroids as a treatment. So it'll be just for a short term around the time of infusion, but for the vast majority of the duration of the trial, it'll be discontinued.
Great, thank you, Kamal. So just a couple of things. Someone is asking about if the patient needs to be on can be on hydrocortisone and prednisone. My understanding is hydrocortisone only, and they can switch to hydrocortisone, I believe, 3 months prior to being entered into the trial. Is that correct, if I remember?
Yeah, that's correct. And we'll work with the treating physicians to hopefully even get that period down, so they maybe could just be on hydrocortisone for 1 to 2 months, but that's correct. Patients just have to be on oral hydrocortisone and not on any prednisone or dexamethasone. And so, if a patient is interested in the study, but they are not on a regimen that is only oral hydrocortisone, then they can speak to their physician, and if their physician thought it was a good idea to switch that patient to hydrocortisone only, then they could, after a couple of months, become eligible for the trial.
Yeah, well, and just as another note there is for the first part of the trial, we're enrolling people who have CAH and are taking only hydrocortisone, as Eric mentioned, but the dose has to be a total amount of 30 milligrams or less per day, because we need to see the effects on your body's ability to make cortisol, and the best way to do that is to be on hydrocortisone and on lower doses of hydrocortisone.
Great, thank you both. So just a couple of things. Someone asked earlier about if the session is going to be recorded. I think Adrenas has to check with their legal team. We will certainly inform you via our website. We also will continue to have information on these trials in our monthly updates and in our newsletter. In fact, we have a Q&A in the newsletter that'll be out in a couple of weeks. If you have any specific questions, you can send them to me, and I will pass them on to the team, and we'll try to get them answered to you. But really, the first step, if you are interested, is to go through the screening process, answer a few questions, and then you can go from there. So I just, on behalf of CARES, I want to thank the entire team at Adrenas for this extremely informative program. I think we had some terrific questions. So I want to thank all the participants. And stay tuned to our website, to our monthly updates, to our newsletter. And I guess I'll turn it over to Eric to say any final words.
Oh, that was terrific. No, and thank you, Dina, and thanks to all of you who joined. We're grateful that you took some time tonight to ask these terrific questions. And as Dina mentioned, please do feel free to get in touch with us through the email address that was up earlier, or as Dina mentioned, if you have questions that you want to email her, she can get them to us. And thank you again. Good night to everyone.
Good night, everyone. Thank you.
Hey, thanks everybody.